Hello everyone, welcome back to our channel The Exploring Medicò. So friends, lastly we had gone through antibody and our today's topic is structure and function of immune system. So I hope it will be beneficial to you all. So let us start our today's topic by introduction. So it is specific organs are responsible for the development and maturation of cells of the immune system which are found throughout the body. There are two types of organs involved with these functions. So these are primary lymphoid organs and secondary lymphoid organs. So primary lymphoid organs are the organs where the lymphoreticular cells develop and matures, and the secondary lymphoid organs are the organs where lymphoid cells and antigens react. Now, types of immune response. So there are mainly three types of immune response: humoral immunity, cellular immunity, and hematopoiesis. So let us discuss one by one in detail. So firstly, humoral immunity. It is also could be said as antibody mediated. So it is immunity mediated by antibodies and produced by plasma cells present in blood and other fluids. Now, cellular immunity. The form of immunity is mediated by sensitized lymphocytes. So friends, I had included this diagram so it could be easy for you, and you can also draw this as it is. Now, hematopoiesis. So all cells of immunological importance originate from a hematopoietic stem cells. So here is the hematopoietic stem cell. So it is basically divided into two major types. So you can plot this diagram as it is easy and also it is important. Now lymphoid system. So the lymphoid system contains two major organs that are lymphoid cells and lymphoid organs. So lymphoid cell contains lymphocytes and plasma cells, and lymphoid organ further divided into primary lymphoid organs and secondary lymphoid organs. So friends, primary lymphoid organs is central lymphoid organs and secondary lymphoid organs are peripheral lymphoid organs. So primary lymphoid organ contains thymus, bone marrow and bursa of Fabricus in birds, and the secondary lymphoid organ contains spleen, lymph nodes, mucosa associated lymphoid tissues. It can be known as MALT in short form, and lymphoid tissue in gut, liver, lungs and in bone marrow. Okay. Now let us discuss these two types in detail. So firstly, central or primary lymphoid organs. So first, thymus. In humans, thymus reaches its maximal relative size just before the birth. It continues to grow until about the 12th year. After puberty, it undergoes spontaneous progressive involution, indicating that it functions best in early life. Now its functions. So it is primary function is the production of thymic lymphocytes. It is the main site for lymphocyte proliferation in the body. Lymphocyte conditioned in the thymus are called thymus dependent lymphocytes or T cells. Second one was bone marrow. So it is the site of B cell origin and development. All lymphocytes originate in the bone marrow. B lymphocyte mature in the bone marrow itself. In birds, the bursa of Fabricus is primary site for B cell maturation, equivalent to the bone marrow. A selection process within the bone marrow eliminates B cell with self-reactive antibody receptor as in thymic selection during T cell maturation. In humans, fetus, Peyer's patches, isolated lymphoid follicles, develops and appears along with cell in the spleen and lymph nodes by the 20th week of the gestation. Now, peripheral or secondary lymphoid organs. So, as we had plotted in the charts, we will discuss one by one in detail. So, first was lymph nodes. So, lymph nodes are distributed along the lymphatic vessels. They contain lymphocytes, that is B and T cell in the cortex, dendritic macrophages, plasma cells, and integrating dendritic cells in the medulla. Now, functions of lymph nodes: it acts as a filter for lymph, each group of nodes draining a specific part of the body, that were phagocytosis, proliferation, and circulation of T and B cells. So, these were the functions of lymph nodes. Now, spleen. So, spleen is the largest secondary lymphoid organ with an important role in immune response. This includes clearance of affected or damaged cells from the bloodstream and host resistance to bacterial infections, primarily directed against blood-borne antigens, mainly pneumococcal and meningococcal infections. So, the spleen serves as a graveyard for non-functional blood cells, a reserve tank for blood, and a systemic filter for tapping circulating blood-borne foreign particles. Now, it's time to discuss mucosa-associated lymphoid tissue, that is MALT. So, they have a rich collection of specialized lymphoid cells like M cells, that is microfold cells, which transport the antigen from lumen of gut to immune cells in specialized aggregates like pair patches or which are scattered. MALT contains lymphoid as well as phagocytic cells. So it contains both lymphoid and phagocytic cells. While predominant immunoglobulin produced in the mucosa is secretory IgA. Now, bone marrow. So it is capable of antigen presentation in an immunological reaction similar to lymph nodes. So that were the classification. Now, cells of lymphoreticular system. So lymphocytes, it constitute about 20 to 40% of the body's white blood cell and 99% of the cells in the lymph. Now, on the basis of their functions and cell membrane components, lymphocytes can be broadly subdivided into three main types, that are T cells, B cells, and natural killer, that is NK cells. So, lymphocytic circulation. So, it is in constant traffic of lymphocytes through the blood, lymph, lymphatic organs, and tissues ensures their contact with antigen in any part of the body. A lymphocyte completes one cycle of recirculation in about 1 or 2 days. Now, functions of lymphocytic circulation. So, mature T and B cells are called navy cells. Their functions are recognition of antigens, storage of immunological memory, and immune response to specific antigens. Now, difference between T and B cells. So, friends, it is very important, so you can note down this, that T cells, it can bind to sheep erythrocytes forming rosettes by the CD2 antigen and CR2 absent, and B cells bind to sheep erythrocytes coated with antibody and complement forming ESC rosettes Due to the presence of C3 receptor on the B cell surface, they do not bind. T cells possesses T cell receptors on the surface composed of two chains of polypeptide linked to CD3 and do not have surface Ig. And in B cell, it possesses immunoglobulin on the surface and have 105 identical Ig molecules on their surface. Now T cells, so it is generally are free of cytoplasmic surface projections, and B cells have an extensively filamentous surface with numerous microvilli. T cells has thymus-specific antigens are present, and in B cells thymus-specific antigens are absent. So in T cell, it undergoes blast transformation on treatment with mitogens such as phytohemoglobin or concaveolin A. And in B cells, it undergoes transformation with bacterial endotoxins, Staphylococcus aureus or EB virus. Okay. Now T cell maturation. So T cell precursors from the yolk sac, fetal liver and bone marrow migrate to the thymus during the embryonic and postnatal stages. So here is the stages of T cell maturation. So you can draw this chart and show the T cell maturation processes in detail. Now T cell receptors. So mature CD8 minus 4 plus TCR alpha beta cells are Hürpel cells, and their functions are induction of B cell differentiation, stimulating proliferating of CD8 plus cytotoxic cells, and production of lymphokinase and regulating certain stages of erythropoiesis. Now types of T cells. So there are various T cells categories that have been identified based on their surface markers, their target cells and their functions. So there are mainly double negative and double positive T cells. And basis of its functional, it is divided into two major types, that is regulatory and effector. Now B cell maturation. So as we had discussed T cell maturation in flow of chart, similarly we will discuss B cell maturation in the flow of chart. So B lymphocyte precursors, pro-B cells, develop in the fetal liver during embryonic life and in the bone marrow afterward and thereafter continuously throughout the life. There are mainly three types of B cells. These are plasma cell, mammary B cell and B1 cells. So you can describe cell maturation in the form of flow chart as I had involved in this diagram. Now null cells, that is about 5 to 10% of circulating lymphocytes, are found to lack features of T and B cells. These are called null cells. Because of their morphology, they are also known as large granular lymphocytes, that is LGL in short form. So their functional characteristics are natural killer cells, antibody dependent cytotoxic cells and lymphokine activated killer. Now phagocytic cells. So these cells are specialized in the removal of foreign and native particles. Phagocytic cells comprise mononuclear macrophages and polynuclear macrophages. First is macrophages. So they may be circulating or fixed cells. It includes monocytes and tissue macrophages. Now macrophages. So these are the polymorphonuclear leukocytes of the blood, and this includes neutrophils, eosinophilic nucleocytes and basophils. Now antigen presenting cells. So it contains dendritic cells, follicular dendritic cells, B cells and Langerhans cells. Now let us discuss abnormalities of immune cells. So abnormalities of the immune cells and the resulting disease are abnormalities or defective mechanisms are stem cell differentiation. So it could be lead to neutropenia, that is very few neutrophils. Then lack of expression of CD18 molecule. It could be lead to LAD, that is leukocyte adhesion deficiency. Then defective phagocytosis, that could be lead to Chediak-Hagessi syndrome. Now defective encoding for NADPH oxidase results in defective intracellular oxygen mediated killing, and that could be results in chronic granulomatous disease leading to life-threatening bacterial and fungal infections. So these were the diseases that could be happen due to abnormality of immune cells. Now classes of proteins. So all the species of animals are found to possess a complex of genes on a segment of one chromosome pair coding for three different classes of proteins. So there are three main classes of proteins. So class 1 protein that determine histocompatibility and the acceptance or rejection of allograft. Class 2 proteins that regulate the immune response, and class 3 protein that include some components of the complement system and few others. Now HLA complex. So the HLA complex of gene is located on the short arm of chromosome 6. It consists of 3 separate clusters of genes. So there are 3 main classes. Class 1 comprising A, B and C loci. Class 2 or the D region consisting of the DR, DQ and DP loci. And class 3 or the complement region, it containing genes for complement components C2 and C4 of the classical pathway, as well as properdine factor B of the alternative pathway, heat shock proteins and tumor necrosis factors alpha and beta. So here is the HLA molecules, mainly class 1 and class 2. So this is the main difference of HLA class 1 and HLA class 2. So HLA class 1 antigens are found on the surface of virtually all nucleated cells, and HLA class 2 are found only on the cells of the immune system, that are macrophagic, dendritic cells or activated T cells and particularly B cells. So HLA class 1, they are coded for by a gene on chromosome 15, and class 2 are coded on the short arm of chromosome 6. Class 1 are having heavy peptide chain, that is alpha chain, and beta 2 microglobulin, that is beta chain. And class 2 has heterodimers with an alpha and beta chain. Class 1 are highly variable amino acid sequences, and class 2 are large nested sets of peptides of variable length. Class 1, they are the principal antigens involved in graft rejection and cell mediated cytolysis, and class 2 are taking part in graft virus host responses and the mixed leukocyte reaction, that is MLR. So thank you friends for listening me, and I hope it will be beneficial to you all. If you like my content, please subscribe our channel and stay updated with our channel. Thank you.